The body protects itself from disease via the immune system. There are three phases to the immune response: physical barriers, nonspecific responses, and then the specific humoral and cell-mediated responses. In the third phase of the immune response, the cell-mediated response is activated and sends out lymphocytes, more commonly known as white blood cells, to help combat an infection. One type of lymphocyte is called a T cell. T cells are first made in the bone marrow and are then transferred to the thymus to mature. All T cells have receptors that they use to recognize foreign antigens that have entered the body. These receptors are proteins that sit on the surface of the T cells, and they have antigen binding sites called epitopes. The epitopes for each cell vary depending on what antigen the T cell is targeting. There can be as many as 10 million different T cells that all target different antigens in the body. It is estimated that each person has enough antibodies to identify 1,000 billion different antigen epitopes. It is through these receptors that T cells are first introduced to the antigen that they will be targeting. Once the T cells are activated, they expand in size and differentiate into various types of T cells. There are 5 types of T cells: helper, cytotoxic, memory, regulatory, and natural killer. The 2 main types of T cells involved with the immune response are cytotoxic, commonly known as killer and helper T cells.
Helper T cells signal the B cells and cytotoxic T cells to start working. In other words, it activates them. They take an antigen presented to them by a macrophage that has consumed a foreign invader. They then bring this antigen to an inactive B cell and present it to the B cell, activating it to help fight the infection by generating antibodies. When the helper T cells attach to a dendritic or antigen-presenting cell, the action causes the release of cytokines that help make clones of the activated helper T cells, all with the same antigen. The cells of these clones release more cytokines that help activate more B cells and cytotoxic T cells.
Cytotoxic T cells target infected body cells, whereas helper T cells help amplify the effects of both cytotoxic T and B cells. All T cells target specific antigens. Once a cytotoxic T cell finds an infected cell, it releases an enzyme called perforin that slowly causes cell death. Perforin, for example, destroys antigens by making many holes in the plasma membrane of the infected cell. T cells make clones of themselves to help fight the antigens in the humors of the body. Cytotoxic T cells secrete interferons, which help cells resist viral infection and help activate macrophages. The interferons also help stop viral replication and enhance the killing power of the cytotoxic T cells. Cytotoxic T cells are most effective against cancer cells, bacterial diseases like tuberculosis that are slow developing, some viruses and fungus, as well as transplanted cells. The ability to recognize all these different antigens is a result of a process called recombination. T cells cut and paste different DNA segments randomly together to create new epitopes for different antigens. The process is intentionally sloppy to allow for random mutations to increase the diversity of the antigens they can target.
The result of the immune system's constant activity protects our body from outside invaders to keep us healthy.